a lot, Tim, for this nice introduction, and thanks, Guy, for having me here today. So today I will tell you something about the main interest of my laboratory, which is the study of the synapse. So since many years, my laboratory is interested in defining what are the processes which control the formation and the function of synaptic contacts between neurons. And in fact, the synapse is one of the most characterized, the best characterized structure in neuronal cells. And there is a presynaptic compartment, postsynaptic compartment, and there are really a huge amount of extremely specialized and well-characterized proteins that mediate on the presynaptic side the fusion of synaptic vesicles with the plasma membrane, and at the level of the postsynaptic compartment, the organization of receptor at the level of the postsynaptic membrane. And there are also transmembrane cell adhesion proteins which sort of keeping register the presynaptic and the postsynaptic compartment. They're really important during development. I will tell you something about those later on. For excitatory glutamatergic neurons, the presynaptic terminals. Can you see me and hear me, Tim, still?
Yeah, all good.
Okay, sorry. I was afraid that something was happening. So the presynaptic terminals are contacting the postsynaptic compartments that are localized in dendritic spines that are protrusions that emerge from the dendrites. And these spines also represents the site where all the processes of plasticity, which are linked to learning and memory, occur in the brain. Now, in the last, let's say, 15 years or so, the interest for the synapse has been increasing even more when it was found that several brain diseases are associated with genes which codify for proteins which are located at the synapse, either presynaptic or postsynaptic compartment. And actually, this was started at the beginning for autism. So for the let's say, the familiar forms of autism, which are really a minority, but the genes which were found to be associated to autism and later on to schizophrenia, so both two important neurodevelopmental diseases, were found to associate to genes that are codifying for protein of the synapse. So neurexin, neuroligins, some of the intersynaptic proteins I've told you before, and for schizophrenia, it occurs for several postsynaptic and presynaptic proteins. So the concept then was extended to additional type of diseases. And indeed, this is one of the schemes that was present in the European Consortium on Synaptic Protein Networks in neurological and psychiatric diseases. And as you can see, there are several genes which has been identified as associated to different type of diseases besides autism and schizophrenia. So epilepsy, measured depression, cognitive disability, and even Alzheimer's disease. So this concept does emerge in literature, the concept of synaptopathy. So the fact that when the synapse is not working fine, is sick in a way, genetically affected, well, this can result in a brain disease. And indeed, a long list of synaptopathy genes have been produced in this last, let's say, 15 years or so. But now we know that neurodevelopmental diseases, like neurodegenerative diseases, are really are complex type of diseases that they involve not only genetics but also environmental type of components. And when I say about environment, I'm saying about the interaction in particular of the nervous system with the immune system. And the fact that the immune system can really play a role in different types of diseases, and in particular neurodevelopmental diseases, may come from the observation, the very solid epidemiological observation, that activation of the maternal immune system during pregnancy is associated with an increased susceptibility for the offspring to develop neurodevelopmental diseases such as autism and later on schizophrenia. And this is further strengthened by the observation that both autism and schizophrenia are clearly associated to an inflammatory type of environment. So potential mechanisms that are linking autism and inflammation and schizophrenia and inflammation have become building up in the last years. So the general idea is that the activation of the immune system, for example, in the mother, like in the case that I was telling you before, can induce the increased production of pro-inflammatory cytokines that can be transferred through placenta or also activate resident immune cells. And this will result in the increase in the production of cytokines at the level of the fetus, which can then influence the neurodevelopment, both in the prenatal period as well as in early postnatal period. And so given all this type of information, in the last years, my laboratory got interested in wondering whether either the cytokines or the microglia, which is also activated during these processes, can impact directly some component of the synapse. But in particular, if the question was whether they can impact specifically some genes which have previously been described to be associated to synaptopathies. Now, in the laboratory, if you want to mimic this kind of situation, so the activation of the maternal immune system and then study what happened to the offspring, the type of model which are most likely, most widely used is the maternal immune activation model, where the pregnant dam is subjected to immune challenges with different type of agents. It could be either poly-IC or LPS or interleukin-6 of different kind of cytokines. And then you can apply this type of immune challenge at different times during development. The most used time windows are GD9, GD15, and then in between GD12, 12.5. And then what you do, you just wait that the offspring is born, and then you observed and analyze the brain of the offspring. And since, we are interested in synapses, of course, so what we do, we analyze the spine density, the formation, and the function of synaptic contacts. Now, if you look in the literature, you find that indeed there is association already for several cytokines that have been found to influence directly some synaptic function. For example, for interleukin-1 beta, it has been demonstrated an alteration on the NMDA receptor-dependent synapses, as well as modulation of postsynaptic GABA receptor. It has been found that interleukin-33 is able to increase the excitatory synapses and so on. Basically, there are indications for several pro-inflammatory cytokines that can affect in general the formation or the function of the synapse. But what exactly about the synaptopathy gene? So can we say that in a way the synaptopathy can be induced or the consequence not only of a genetic type of dysfunction, but can result from an environmental type of dysfunction? So a few years ago, we started investigating the pathway of interleukin-1 beta, and to investigate the possible upregulation or downregulation of the interleukin-1 beta pathway, we used the interleukin-1 receptor antagonist, anakinra, which is a blocker of the interleukin-1 receptors, or to potentiate to see what happens when the interleukin-1 beta pathway is enhanced. We took advantage of a mouse model that was developed in Humanitas by Alberto Mantovani and Cecilia Garlanda, which is the interleukin-1 receptor 8 knockout mice. The interleukin-1 receptor 8 is a receptor that is dampening, is sort of blocking the activation of the interleukin-1 receptor. So while with anakinra, you get a downregulation of interleukin-1 beta pathway, with the interleukin-1 receptor 8 knockout mice, you get an enhancement of the interleukin beta pathway. And what we found is that if you apply protocols for inducing long-term potentiation in wild-type mice and wild-type neurons, you are able to induce the what is expected, the increase in the density of mushroom spine and the increase in the electrophysiological property in the frequency of miniature events in the wild-type neurons, both if you downregulate the interleukin-1 beta pathway, or if you upregulate the interleukin-1 beta pathway, you are completely getting to a block, impairment of the processes of long-term potentiation, both by counting the synapses and measuring the electrophysiological activity. But if you take the interleukin receptor knockout mice and you apply anakinra, just blocking the excessive activation of the receptor, you can rescue the potentiation phenomena. We try to analyze what are the molecular mechanisms for that. And to cut a long story short, what I want to tell you today is that we end up picking MCP2. And we found that indeed the expression of this gene, MCP2, is increased when interleukin-1 beta pathway is potentiated, and it is downregulated upon blockade of the receptor. Now, MCP2 is a transcriptional repression, and the target genes are several genes of the synapse. And MCP2 is a specific gene that is involved in both the Rett syndrome, when the gene is lacking, and in the MCP2 duplication syndrome, when the gene expression is excessive. And both these diseases share mental retardation, behavioral problems, autism, and so on. And we were able to demonstrate that indeed MCP2 is causal in this type of defective phenotype, because by downregulating the expression of MCP2 in the interleukin-1 receptor 8 knockout mice, we were able to restore the phenotype. So this was a first indication that indeed the pro-inflammatory cytokine can modulate the expression of one of the most relevant genes of synaptopathy, MCP2. More recently, Filippo Mirabella and Davide Pozzi started investigating instead what happens in the model of maternal immune activation. And so what they did, they injected interleukin-6 in the pregnant dam and went analyzing the brain of the offspring at P15. And what they found, that upon this type of treatment, there is a significant increase in the amount of synapses, excitatory synapses that are formed in the brain of the offspring, just the excitatory, not the inhibitory. And here you see excitatory functional synapses, nothing the inhibitory ones. So what we did by single cell RNA-seq to get an idea of what could be the pathways that are involved in that. And we found that indeed, downstream of interleukin-6, there is STAT3, and in fact, there is a STAT3 activation specifically in the excitatory pyramidal neurons. But when we went looking to the differential expressed gene, we found just few of them. But the very first one, the most relevant one, which is as well upregulated exclusively in the excitatory neuron, is RGS4. But RGS4, again, is a synaptopathy gene. It is in particular one of the top candidate genes for schizophrenia. And again, we had the possibility to demonstrate the causal relation between the defective phenotype, because by using blockers of RGS4, there are luckily small molecules that are blocking this molecule, we were able to restore the defective phenotype. So again, is a cytokine interleukin-6, in this case, prenatal immune activation, we have a target gene, which is a synaptopathy gene. And this is not just from our laboratory. From the Pletnikov lab, for example, they found an interaction between poly-IC and DISC, which is a synaptopathy gene which is involved in schizophrenia. Last year, Camilla Bellone's lab, a very beautiful paper in which she found an interaction between LPS and SHANK3, which is one of the most relevant genes which is involved in autism. And also LPS, ECDL lab, 2013, LPS and alteration of the intersynaptic protein, neurexin and neuroligands. So from all this type of evidence, we can say that indeed, yes, the answer is yes. The activation of the immune system under different type of condition is in fact able to specifically hit synaptopathy genes. But of course, when we talk about the development of the brain in the prenatal and early postnatal period upon the activation of the immune system, so far I've been talking about the cytokines and the soluble factors. But of course, we know very well that the microglia is on the first line. And so we wondered whether microglia could be also involved in the homeostasis of synapses in periods which are more related with neurodevelopment. And this is very much possible because we know that microglia is able to induce a synapse formation and is also able to perform a process of synapse elimination. We know very well that branches, axonal branches and synapses, which are less active, are eliminated by the axon pruning and synapse elimination processes that are performed by microglia. And we also know something which is really interesting. A dysregulation of these processes, both the formation, but in particular the elimination of synapses, if there are defects, dysfunction in this process, this can associate to neurodevelopmental diseases. And in particular, an excessive elimination of synapses is linked to schizophrenia, whereas a defective elimination of synapses is linked to autism spectrum disorders. So in our lab, we started looking and asking whether microglia could really play a role during the neurodevelopmental window. And we got interested in one protein that is very well known by everybody that works with microglia, which is TREM2. And TREM2, you know very well, which is a cleaved player in microglial biology, can be cleaved by ADAM17 or ADAM10 to produce a soluble form of TREM2. And quite interestingly, it is strictly associated with the risk of developing Alzheimer's disease. From the work of several laboratories in the last year, the general concept is that TREM2 is important for mediating the shift from the stage 1 DAM to the stage 2 DAM, thus making the microglia able to respond properly to the damage. And in the absence of TREM2, this process would not occur in a proper way. And also we know that TREM2 is able to have proliferative and pro-survival functions through PI3 kinase and mTOR pathway, and it is also able to modulate the metabolism of microglia. So it is a very central molecule, which is largely investigated in the later stages of development, so aging and Alzheimer's disease. But we decided to take a look to TREM2 during neurodevelopment. And indeed, a few years ago, we were able to demonstrate that TREM2 is indeed required for the process of synapse elimination. In particular, what we did, we looked at synapses in TREM2 knockout mice at P20. So we moved to really early neurodevelopmental window. P20 is the period when the elimination of synapses occur at the level of the hippocampus, which is our favorite area of investigation. And what we found is that if you look to either presynaptic proteins or postsynaptic proteins, you can see that the TREM2 knockout mice display an increased density of synaptic contacts at P20. And these contacts are functional because if you record acute slices by electrophysiology, by patch clamp, you can see that they are characterized by significantly higher frequency of miniature excitatory events. And of course, in perfect line with that, even if you stain by Golgi staining the hippocampus, you can see that the number of the density, let's say, of dendritic spines is higher in the CA1 hippocampal pyramidal neurons in TREM2 knockout mice as compared to wild type. So, okay, this is interesting, but we have no idea of why is that. So in order to get some information about the reasons which may lead to this increased density of synapses at early developmental stages, we started growing in co-culture wild-type neurons together with microglia, either microglia from wild-type mice or microglia from TREM2 knockout mice. You can keep them in culture for 24, 48 days. And if you do that with the wild-type microglia, you can see that after 24 hours, you can already appreciate a reduction in the density of synapses that are formed by these neurons that you can detect in these neurons when wild-type microglia is co-cultured. But if you put in culture instead of wild-type the knockout, TREM2 knockout microglia, not even after 24 or 48 hours, you do not see any reduction. And again, everything was confirmed by electrophysiology. So reduction by wild-type microglia, no reduction with TREM2 knockout microglia. So, okay, this could be, of course, possible, raise the possibility that the TREM2 knockout microglia is not able to properly to eliminate and phagocytose synapses. But of course, this is not a demonstration because the formation of synapses could be differentially affected. So to address more in detail, we did the synaptosome preparation, which is something that is done routinely by people that are working on synapses, and we label the synapses by these synaptosomes. So these are isolated synapses. There is a presynaptic terminal that contains a synaptic vesicle and mitochondria, and usually there is a part of the postsynaptic density which stay attached to the synaptosome. So it's a preparation where you have a lot of presynaptic, but also postsynaptic compartment. You can see from this reconstruction in this paper. This is not our paper. But so we labeled this synaptosome with FM143, and we fed either wild type or TREM2 knockout microglia with the labeled synaptosomes. And as you can see is that after 2 hours or 24 hours, the knockout microglia, which is the purple one, is significantly defective in the engulfment of labeled synaptosomes, thus indicating that indeed in vitro, microglia is less, from TREM2 knockout mice, is less efficient in the phagocytosis of synaptosomes. But of course, again, we are in vitro, so we needed to see that in vivo situation. And so we moved to the real brain and we analyzed the amount of synaptic material which is engulfed inside microglia of either wild type or TREM2 knockout mice, and we found a significant reduction of synaptic material engulfed in the CD68 positive phagolysosomes inside the microglia, thus indicating that TREM2 is indeed required for the correct synapse elimination during development. Now, what are the mechanisms which really mediate the recognition of synapses that are to be eliminated? We know from the beautiful work of Dorothy Schaffer, Beth Stevens, and several other laboratories in these years that ITMI signals are probably labeling the synapses and the axons that need to be eliminated. And probably they are the weak synapses, the less active synapses and axon, which become tagged with these ITMI signals. And so they become sort of flagged for being eliminated. And among the ITMI signals, of course, the most characterized and famous one, well demonstrated by Beth Stevens, are the complement components, C1q, C3, that can be recognized. They label the synapses, the weak synapses, and are recognized by receptors that are present at the level of the microglia. So we wanted to understand whether there are additional components, because as you can see here, we have TREM2, but we didn't know exactly which was the possible receptor recognized on the synapses. And so a couple of years ago, we started this very nice collaboration with Beth Stevens' lab, in which we wanted to understand whether phosphatidylserine could be a putative binding or ITMI signal for the microglia-dependent process of synapse elimination. And this was, and I mean, we were interested in that from the TREM2 side, because Marco Colonna has nicely demonstrated that TREM2 is able to bind lipids and also phosphatidylserine. And we know that phosphatidylserine is usually inside the plasma membrane, but can be translocated on the outer surface of neuronal or in general cell membranes and represent a signal for apoptosis. And we also know that there are tools such as Annexin 5, which is able to specifically label the externalized phosphatidylserine. So we gave a try, and we tried to labeled by Annexin 5 primary cultures of hippocampal neurons. And indeed, we found the colocalization with postsynaptic marker, presynaptic markers, in some cases, very nice labeling of dendritic spines. But of course, these are primary cultures that could be, and this could be just the result of a degeneration of neuronal death. So we needed to make one step forward and do this kind of experiment in vivo. And this is what we did, and we took mice at P10. Again, we are right in the middle of the period of synapse elimination into the hippocampus, and we took advantage of a different dye, which is called PSV. It is a fast calcium independent dye. It is able to bind to phosphatidylserine, only externalized phosphatidylserine. It is completely cell impermeable and is small in size, so it has all the features that are required if one wants to see whether phosphatidylserine is exposed in ex vivo to colocalize into with synapses. So we did these experiments, and we used Bassoon as a presynaptic marker and PSD95 as a postsynaptic markers. And at P10, we quantified the amount of colocalization of PSV with the presynaptic or the postsynaptic markers. And we found that PSV, is able to colocalize, but it colocalizes much more importantly with the presynaptic markers as compared to the postsynaptic one. And then we did the same experiment at different time windows. And what we found we thought that was really interesting is that the exposure of phosphatidylserine was the highest at P10, and then it was progressively decreasing. And at P30, when the process of synapse elimination is at the level of the hippocampus, is completed, is finished, there was no exposure of phosphatidylserine. But what was really exciting is that Nicole and Beth did exactly the same type of experiment on their favorite area, which is the lateral geniculate nucleus. They used different markers. They used VGLU2 for the presynaptic and Homer for the postsynaptic. These are the biomarker, I mean, the markers, the synaptic markers, which are more heavily expressed at the level of the lateral geniculate nucleus. But as you can see here, it is impressive that, again, even in the lateral geniculate nucleus, there is a colocalization of PSV more with the presynaptic and less with the postsynaptic compartments. And then also Beth did the experiments during development. But of course, here the time of development of the lateral geniculate nucleus is earlier as compared to the hippocampus. And indeed, the highest exposure of phosphatidylserine was at P5 and was completely zero at P10 when the process of synapse elimination in the visual system is almost finished. So the exposure of phosphatidylserine, in a way, is able to correlate with the timing of the process of synapse elimination in two different brain areas. To then make an example and make sure that indeed the ability of microglia to phagocytose synaptosomes or anyway liposomes containing phosphatidylserine, that really phosphatidylserine is crucial for the ability of microglia to phagocytose. Well, we started the collaboration with Rainer Jan lab, and he produced for us liposomes with different amount of phosphatidylserine as compared to cardiolipin. So we had this liposomes with 90% of phosphatidylserine or with 20% of phosphatidylserine. And then we fed the microglia with these liposomes with different amount of phosphatidylserine, 99 or 50%. And what we found is that wild-type microglia is progressively reduce its ability to phagocytose liposome in parallel with the reduction of phosphatidylserine into the liposome membrane. And it was really interesting for us, the fact that if you do the same with knockout microglia from the TREM2 knockout mouse, there is no really difference on the presence of different amount of phosphatidylserine. There is some phagocytosis, which is the typical fluid phase phagocytosis, but no dependency on phosphatidylserine. So this data indicated that besides the complement component, also TREM2 and the phosphatidylserine, but also other components maybe besides TREM2. This is, for example, another molecule which has been identified as a binding, as being able to bind phosphatidylserine, thus indicating that probably the process of elimination of synapses need to be really well controlled. Just one type of process is probably not enough, and we need to have a different and multiple levels of control in order to make sure that the defective synapse is eliminated. Now, in the last months, we have been curious, we started to be curious to understand whether there are also additional binding proteins for TREM2. So again, we prepared the synaptosomes from brain at different developmental stages, and then we took advantage of a recombinant protein, which is formed by the Fc region of the human EGG, fused with the extracellular domain of TREM2. And we used this Fc TREM2 as a bait to isolate putative TREM2 interactors from synaptosomes that we have been isolating, as I told you, from different time of development. So it's something that we are doing. So I do not have any real result to show, but what I can tell you that I think is interesting is the demonstration and the further confirmation that indeed for TREM2, the binding proteins that have been identified by mass spectrometry analysis, they are mostly localized at the presynapse. And if you look, you can, and if you look, you can see both extrinsic proteins of synaptic vesicle membrane, presynaptic membrane. And if you look to the type of disease that are associated, you see intellectual disability, you see seizure, you see epilepsy. So it seems to be really interesting type of approach, and we hope that we will be able to identify something in the next future. And in particular, we are curious to understand whether we can see something different during development in the adult, and why not even during the aging. Now, everything that I told you for this ability of microglia and the TREM2 defect in the pruning of synaptic contacts is something that we have been observing essentially at the level of the CA1 region of the hippocampus, okay, where you have more synapses and that are more active at P20, period of synaptic pruning. But if you look the same in a quite close region, which is the CA3 region of the hippocampus, P20, TREM2 knockout mice, we don't see any difference, not in the synaptic density, not in the activity. So this means that there are differences, regional differences in this property of microglia, and in particular of TREM2 to be involved in this process. And we don't know what is the difference, but since there is a heterogeneity of microglia in different areas, it is possible that the microglia can have different type of feature. Of course, this is something well expected from the CA1 and the CA3 region. And we started taking a look to the expression of TREM2, and we found that despite the similar number of microglia and the IBA1 volume in the two regions, the expression of TREM2 is lower in the CA3 region as compared to the CA1. So we are now going on in order to understand whether this is at the basis of this difference. But something that we found even more intriguing is the fact that this increased density of synapses is something that you see in the CA1 region at P20. But what happens at later developmental stages? So we did the same kind of analysis at P90, so early adult mice. And what we found is that at P90 in the CA1 region, the density of synaptic contacts in TREM2 knockout mice is significantly lower. So it was higher at P20 and become significantly lower at P90. And of course, the CA3 region, it doesn't care at P20 and it doesn't care at P90. So this is something that is occurring specifically in the CA1 region. Of course, we want to understand what are the mechanisms, what's going on here, why this increased number of synapses is then followed during maturation by a significant reduction. So we have been looking, Rafael in the lab has been looking to different types of processes. We look to the CA1 thickness, so we checked for the possible neuronal death. We check for the possible reduction in the expression of synaptic vesicle proteins. We didn't find anything. So we reasoned, well, maybe the ability of microglia to phagocytose the synapses is defected at P20, but something happens at P90, a compensatory mechanism. But again, even at P90, the microglia is characterized by a lower ability to phagocytose synaptic material. Of course, we cannot exclude the possibility that astrocyte could take the stage at this point, but it's something that we are analyzing right now. But what is interesting is that if you look at the morphology of these synapses in the CA1 region of P90 mice, and this is how the phylopodia become then differentiated in mushroom, so that the mushroom are let's say the fully mature type of spines, whereas the phylopodia are the immature type of spines. What we found is that indeed in the CA1 region at P90, there is an increased higher abundance of phylopodia as compared to the wild type. And Erika Tagliatti, in collaboration with Katia Cortese, did electron microscopy characterization of these synapses, and she found some defect in the synaptic area, something that she's now more, analyzing more in detail. But you can see already that the synaptic area is significantly lower in the CA1 region and not in the CA3 region, thus suggesting that these synapses in this area are the sort of maintain a sort of immaturity, suggesting that a failure to eliminate synapses at the correct developmental time may prevent the normal maturation and the strengthening of synaptic connections. And this is something that we would like to univocally demonstrate in the next, possibly next months. But now, what are I've told you about the synapses. P90, young adult, we have a reduction in the synapses, so something wrong happens. But what about the behavior, for example, of these mice? Do the TREM2 knockout mice behavior in a normal way? So we started to look at their behavior in young, very young mice. And as you can see here, the juvenile play, which is just a way of measuring the reciprocal interaction of these young mice, is lower in the TREM2 compared to wild type. And also, this is sort of mirrored to what happens in the adult, in which if you have an adult mice and you make it choose between an empty cage and the cage which contain a co-specific mice, the wild-type mice spend normally more time close to the cage which contains the other mice, because it's a sociable but is a social type of animal. But if you have a TREM2 knockout mice, well, this sociability seems to be defective. So they display defects in sociability. And also the TREM2 knockout mice, they also display an increased type of repetitive type of behavior, such as, for example, the self-grooming. Now, all this type of behavior are normally generalized, seen as autistic-like behavior. But then we reason, okay, this is behavior, but what about the circuits? Because between the synapses and the behavior is a very long step. So let's try and see what happened with the circuits. And so in collaboration with Valerio Zerbi, that was at the time at ETH Zurich, he did resting state fMRI experiments in wild type and TREM2 knockout mice, young again, 2 months old, more or less. And what we found is that the TREM2 knockout mice displayed defective connection, reduced connection between hippocampal areas and notice CA1 area in particular, and the prefrontal and the retrosplenial cortical areas. So this is very interesting because now we have an alteration of synapses. We have something in the circuits that is not working, and we have behavioral defects. But is this something which is related, this defects in circuits, is it something that could be related to autistic-like features? And the answer comes from this paper that was done last year, from a couple of years ago, from Valerio Zerbi, together with Alessandro Gozzi and Nicole Wenderoth. We were collaborators in this paper. And this is a very nice paper because it stems from the problem that if you are interested in autism and you want to understand what are the connectomic features of autistic brain, well, it's a mess, because if you go into the literature, you find everything and the opposite of everything. In some case, you find hyperconnectivity, underconnectivity. So what Valerio and the collaborators did, they took 16 autism mouse model, okay? The most established mouse model of autism. There are 16, and they examined the connectivity properties, features of all these mouse models. And what they found is a continuous connectivity landscape. But what was really interesting is that they were able to segregate all these connectivity features in 4 different clusters. And this is something that were characterized by completely different connectomic landscape. And so this is important for people that work in autism, because it can provide an explanation to the fact that sometimes in some models you see hyperconnectivity in certain areas, and in other models you see underconnectivity. And of course, if you are looking to different models, it's something that you do expect. But for us, for TREM2, was really exciting for us to see that the connectivity of the TREM2 knockout mice was going in the same, was completely superimposable with the connectivity of one of the most established autism model, which is the FMR1 knockout mouse, which is the model for the Fragile X syndrome. So in particular, both the TREM2 and the FMR1 knockout mice, they were shared the similar defects in connectivity, under long-range connectivity between cortex, striatum, and thalamus, and over-connectivity inside the short-range connectivity inside the hippocampus and the hypothalamus. And so this tells us 2 really important issues, that in fact, it is not just the fact that we have more synapses, defective pruning, like it occurs in autism, that we have behavior that is similar to what we have in autistic mouse model, but we also have a connectivity type of feature in the TREM2 knockout mice, which is superimposable to those of the Fragile X. And of course, you would ask, what is Fragile X? Well, you know very well that the Fragile X is an RNA binding protein, which is highly expressed in the brain. And it is important because it controls the translation of several proteins, which are important for the synapse. but apparently it doesn't anything to do with microglia. So we are curious to understand why it's that. It could be that just the TREM2 knockout mice and the FMR1P knockout mice, they display the same deficit of synapses or the same increase of synapses in the same area. We don't know that. But in the meantime, we are taking a look to the microglia of these mice, and we found something interesting that, so this deserves a further investigation, that in the knockout mice, the FMRP knockout mice, there are defective in the morphology of microglia, even if we know that morphology says not that much, but anyway, there is an alteration in the morphology of microglia. But what is interesting is that this occurs exclusively at P20, and it doesn't occur, it's completely reversed and normalized at P40. And at P20, we also have a significant decrease of the messenger for both TREM2 and for ThyroBP. So this Antonella Borreca is just curious to see whether it could be, if there is, and which one could be the connection between the TREM2 knockout mice and the FMR1 knockout mice, which can provide an explanation to these completely shared defects in connectivity. And as a last demonstration for the possible involvement of TREM2, even in neurodevelopmental diseases, is this analysis that we have done in postmortem in brains of postmortem patients, and we examined the amount of the protein in the postmortem brain, and we had a group of patients going from 2 years of age up to 65 years of age. And if you look at the amount of TREM in all of them together, you do not see any difference. But if you focus in the group of patients that collected the brain that are in the period of synaptic pruning, which in the humans is between 2 years of age and 20 years of age, then we have a significant reduction in the levels of expressed TREM2. And also, if you make a correlation between the levels of TREM2 and this parameter is called ADIR, which is an autism diagnostic interview, you see that there is a correlation between the levels of TREM2, this time in the entire population, and the, let's say, how much relevant is the disease in these patients. So whether this is causative, we don't know. Why could be that we have such type of reduction? Also, we don't know. So far, I can tell you that there is no connection described, any association described in literature between TREM2 and autism. But there is this reduction, which is interesting. And something that is also interesting is that we know that the levels of TREM2 can be reduced by inflammatory type of stimuli. We have done something with poly-AC, but even more interestingly, Elisabetta Menna did that in collaboration with Stipan Ionic in Rijeka, and is an expert of cytomegalovirus. And as you can see here, in young mice that are infected with the cytomegalovirus, there is an enormous reduction of the protein. This is the messenger, but this one is the protein in the treated mice. Thus suggesting that indeed even environmental type of stimuli can reduce the level of TREM2, and the level of TREM2 seem to associate in a way with some defects that can be found in autistic patients and autism models. So in conclusion, we started studying neurodevelopmental diseases such as autism with a complete view focused on genes. And in 2012, we were saying genes linked to the spectrum of disorder with pathways, and synaptic pathways in particular have been found to gain ORI in autism. But now we know that, in fact, the same type of defects can also result from different type of processes which directly involve the immune system. The microglia, on a way, so pointing to the crucial role of the synapse elimination during development in the autism spectrum disorder type of phenotype, and as I showed you in the first part of my talk, in the cytokines, which may specifically affect some of the protein which has been previously identified as a synaptopathy genes. And for this reason, we have decided to try to coin this term, which is called immune synaptopathies, meaning the activation or the alteration of the function of the immune system, which specifically hits the synapse and more in detail synaptopathy genes, and can then result in pathological and neurodevelopmental type of diseases. And I just wish to thank the people in my lab. In blue are the TREM people, in black are people that are working on different projects that they didn't have time to talk about today. These are our collaboration, Davide Pozzi in particular and Jenny Desiato, who are really strong collaborators in this process, but also Simona and Sara Mancinelli, Elisabetta, as I told you, and our external collaboration with Rosa Paulicelli, Beth Stevens, Marco Colonna, Fabia Filippello, now she's back after a period at Washington University. And then our most recent collaboration for these two ARANET neuron projects that actually we are in particular recruiting from one of that is called the microblitz. And these are our grants. And in particular, I wish to tell that we would keep in mind that we are hiring. So if someone is interested in joining the lab, we are ready to get you.